Hello everyone, my name is Yanshole and I am a scientist from the International Tomography Center here in Novosibirsk, and thank you for the opportunity to tell you today about metabolomic profiling of biological fluids of patients with lymphedema. Despite the high diagnostic level of modern medicine and modern medical technologies and instruments, there are still some diseases that are not easily diagnosed and prognosed. One of them is lymphedema, or so-called elephantiasis. This is the accumulation of protein-rich fluid in extracellular space under skin and subcutaneous tissue. Lymphedema is an external or internal manifestation of lymphatic system insufficiency and deranged lymph transport, causing the chronic inflammation in human body, adipogenesis, fibrosis, and as a result, the increasing of volume and disfiguration of upper limbs, arms, and lower limbs in men and women, in women especially. There are two causes of lymphedema. One of them, this is a congenital lymphatic dysplasia, also called primary lymphedema. This is a very rare case. It's just one patient per 100,000. And today we will talk about secondary lymphedema. This is acquired obliteration, such as after radical operative dissections. Here on this slide, I should show you not very pleasant, but very important pictures that show stages of lymphedema. The first stage, where we can see damaged lymph vessels, but no clinical manifestation, with the early and not very high accumulation of protein-rich fluid when the edema is pitting. The second picture, this is the second stage, which divides to two parts. This is the early stage, when the protein-rich fluid in interstitial accumulates, but edema is still pitting. And the later stage, when the lymph doesn't pit because of excessive subcutaneous fat and fibrosis. And my samples for today, this is the stage 2 of lymphedema in women. And the last stage, very important, this is the stage 3. This is very dangerous stage with chronic inflammation in human body, with persistent accumulation of metabolism products and further deposition of fat and fibrosis and warty overgrowth development. I should say that the most important reason of lymphedema is breast cancer. And here I show you some statistics. So you can see that about 20% of lymphedema after breast surgery is the axillary lymph node dissection, about 6% is the sentinel lymph node biopsy, and any type of regional nodal radiation may lead to lymphedema. This is an increased risk of lymphedema in future. This is foreign statistics, and what we can say about Russia and Novosibirsk particularly. Our colleagues in the clinics of the Institute of Clinical and Experimental Lymphology have patients with lymphedema. This is mostly women, which have breast cancer surgery and radiation after in their anamnesis. So this all speaks in favor of relevance of this research for the world science and for the Novosibirsk particularly. This is a very important phrase that was published by the Executive Committee of the International Society of Lymphology last year, and it sounds like research in molecular lymphology, including lymphatic system, genomics, proteomics, metabolomics, and systemomics should be greatly expanded. It means that there is some information about genetics of this disease. There are papers about proteomic of the interstitial fluid in lymphedema, but there is still lack of information about metabolites and metabolomic changes during lymphedema development. And it means that the systematic information combining genetic information, proteomics, and metabolomics may be very important for understanding the etiology of this disease. here I show the pictures from our colleagues from the Laboratory of Operative Lymphology and Lymphatic Detoxification here in Novosibirsk. And on the left-hand side, this is an ultrasound examination of lymphedema in arm after breast cancer treatment. And here you can see the non-affected lymph. And on the left-hand side, this is a affected lymph, where here with black, we can see the accumulation of free fluid in the subcutaneous tissue. Also here, we can see in dark the cavities which contain free fluid that accumulates in edema, and this is called lymphedema. Sometimes it is possible to extract this free fluid from patients, not from every patient, but sometimes it is possible. And how it is look like? So you need to be very precise to get into this cavity to extract interstitial fluid without mixing with blood, because our research is aimed to compare human serum with human interstitial fluid to make it possible to find some unique or complex biomarkers for prognosis and diagnosis of lymphedema with metabolomic approach. Some words about metabolomics and metabolites. Metabolomics, this is a young branch of post-genome sciences. So metabolomics is about small molecules under 1,000 Daltons. This is amino acids, antioxidants, nucleotides and nucleosides, organic acids, lipids, sugars, and osmolytes. There are different approaches in metabolomics. So the not very interesting one, this is a qualitative metabolomics, when you can say that sample A contains alanine, for example. The more interesting is semi-quantitative approach, when you can say that content of alanine in sample A is twice as large as in sample B. And the most interesting one, this is a quantitative approach, when you can define the exact concentration of alanine per gram of tissue or per milliliter of biological fluid. So you can get the metabolomic profile of, for example, normal tissue or fluid, compare it with pathological metabolomic profile of tissue or fluid, find some metabolites that are different in these two cases, and then looking on the map of metabolic pathways, you can find these metabolites, for example, here, here, or here, and say that changes in concentrations of the metabolite here may lead to changes in concentration of metabolite here that ends with the changes of the concentration of end product. Another way and another approach, metabolomic approach, is the comparing of pathological tissue or fluid with pathological tissue or fluid treated with drug. So in this case, you can say which metabolic cycles and pathways were disturbed, which pathways are affected by the drug, and is it possible to back the metabolomic profile of pathological tissue after drug treatment to normal one. In our case with lymphedema, we will compare pathological human serum with pathological human interstitial fluid to make an attempt to find some kind of biomarkers for prognosis and diagnosis of this disease. We have these samples from one patient, make the metabolomic extraction, have the extract, and then we have two ways how we can analyze this. NMR method is the quantitative one, very straightforward, when you can calculate the concentrations of most abundant metabolites in fluid or tissue. For now, it is made in this way is made, so we measure the NMR spectrum of serum and interstitial fluid, and the second way is HPLC-MS analysis that is not made to now, but in this way you can analyze hundreds of metabolites in semi-quantitative mode. And this some preliminary results that we have. So this is the representative NMR, proton NMR spectrum. All lines are annotated with our homemade database, so we can say which line corresponds to which metabolite. And this is the results in concentration. So we measured about 40 metabolites in both in serum and interstitial fluid of one patient. And at first, I should say that in this patient, we found a very high level of glucose. And in green, I marked the acids, organic acids, which concentration in interstitial fluid is higher than in serum. So, and in yellow, the acids that which concentration in serum is higher than interstitial fluid. And for example, these metabolites may serve as potential biomarkers for prognose the lymphedema in patients. But to be confident, we need much more statistics to make the very good results. And our colleagues from the Laboratory of Operative Lymphology and Lymphatic Detoxification, they have some more samples of serum and interstitial fluid, and this work will be continued soon. I think till the end of the year, it will be finished. Thank you for your attention.